Good evening, everybody. Thank you for joining our webinar. It's Pancake Day. I'm sure you've all enjoyed a pancake and that you'll be able to learn in the next hour and a half about the effect of that food on your microbiome. We're delighted to welcome two very eminent speakers, Falk Hildebrand from Norwich, and our own committee member, Kevin Barrett, who's a GP in London. I'm Marion Sloan. I'm current chair of the Primary Care Society of Gastroenterology, and the PCSG has worked with the Quadrum Institute to offer you 4 of these webinars all about the microbiome. I think there's intense interest about the microbiome, and today's evening offering is about the microbiome and stress. And then we've more still to come: IBS, aging and immunity. So during the year, we'll be able to offer you insights into this very new and evolving area of science. The evenings have been hosted by Quadrum Institute and provided by Alfric's PrecisionBiotics in an unrestricted educational grant. And we're very, very grateful that we're able to bring you quality education like we're going to have this evening. So each speaker will introduce themselves. The plan is to go straight from one talk to the next, and then we'll be very grateful for questions at the end. Please may you put them in the Q&A, and we'll try and group groups of questions together so that we can give you some meaningful answers from our speakers. So thank you for attending. We're delighted that you're here. And it gives me great pleasure to introduce Dr. Falk Hildebrand to give us the first talk. Thank you.
Yeah. Thanks a lot, Marion. Let me just switch over to my slides. So you should all be seeing now my slides, and especially my first slide here. Yeah, it is really a pleasure to be able to speak to such an audience today virtually. And today I want to talk a little bit about the research I'm doing at the Quadrum Institute. So my name is Falk Hildebrand, and I will be talking about gut feeling, how the microbiome shapes our well-being with a specific focus on diets. But just a bit of background about myself. So as you might have guessed from my accent, I'm a German. So I did my undergrads in Constance. Where I did a bachelor in biology. I moved then actually afterwards to Brighton so to do my master in the evolution of bacteria. After that, I moved to Brussels. Here's where I started working with gut microbiome specifically for my PhD thesis. And then I did my postdoc in Heidelberg in Germany again, continuing to work on gut microbiomes. With this research, I then moved to Norwich to lead my own group here. That was 6 years ago already. And this research is again about the gut microbiome, but of course different aspects to it. And during all this time, I think one of the driving questions was for me, what is a healthy gut microbiome actually, and what does this mean for us? But let me start a little bit more at the beginning. So being a German, of course, I also have to include a German slide, but don't worry, it's the only one during this talk. So this is actually about, let me just say that it says the wonder of the gastrointestinal lining, right? So, and it says here, I hope you can see my mouse, on a single square centimeter on intestinal lining, there are 3,000 intestinal villi, of which each is covered by 3,000 cells, of which each carries 100 microvilli, these little parts here, right? And just as a comparison, gut bacteria are roughly this size compared to the human cell that line now our intestine. So it's very, very small cells. But we do know about them for quite a while already. In fact, in 1681, Antony van Leeuwenhoek, the inventor of the microscope, you can see his invention here on the left side, he had diarrhea apparently, and so he notes down in his diary, my excrement being so thin, I was at diverse times persuaded to examine it, wherein I have sometimes also seen animalcules are moving very prettily, some of them bigger, others a bit less than a blood globule. And that's a very exciting discovery he makes here. So on one hand, he's talking about animalcules, And the reason why he uses this word is because bacteria as a term, as a concept, didn't exist back then. Yeah, but he discovers that there's little things moving within his stool sample. So this is the first discovery actually of the gut microbiome. Nowadays, we now understand a lot more about these bacteria that are colonizing our human gut. Here you just have a graph on the left where you can see how many microbes per milliliter we would expect. The stomach being pretty much aseptic, so there's not many bacteria there. And then the mass or the load of bacteria just increases more and more until the colon, where you have 10 to the power of 11 to 10 to the power of 12 microbes per milliliter. So an immense density of microbes. It's one of the most densely colonized microbial environments that we actually know of is the human colon. Overall, every one of us carries roughly 200 grams of live bacteria with them all the time, with fluctuations during the day, of course. And in general, you can say there's probably 10 times more bacteria than human cells. Yeah? And we estimate that each one of us has probably 200 microbial species. And altogether, these 200 different species carry now 1,000 more genes than we actually have genes encoded in our own genome. So it is a really rich and densely colonized ecosystem that is in every one of our guts. Now, what is this actually good for? It's actually quite important to us. We understand this now. For example, the human gut microbiome is very important for breaking down fibers that are otherwise indigestible by us. It's also very important for providing us with vitamins and cofactors that we cannot produce ourselves. Early in life, very important for the maturation, late in life for the modulation of our immune system, and something that's often a bit overlooked, the gut microbiome, the bacteria here, form a first-line defense against invading pathogens. Yeah? So without this protection, we would probably have enteric diseases much more often than we luckily mostly don't have. And many more associations are also found with time. So we do know that the gut microbiota and the human host, there's a lot of interactions going on, and we understand there's a lot of diseases by now that are somehow associated to imbalance in the gut microbiome, right? So if there's an imbalance, usually we talk about dysbiosis, especially if we think this is related to diseases. What kind of diseases are there that we think are influenced by our gut microbiome? Well, on one hand, we have for a lot of brain-related diseases, so CNS-related disorders such as Alzheimer's, Parkinson's, but also depression, cardiovascular diseases, liver diseases like NAFLD, infection diseases, enteric diseases. A classic among these is IBD, inflammatory bowel disease, horrible disease, but definitely associated to dysbiosis in the microbiome. But also cancers like colorectal cancer, for example. We do know that there, in some cases, there is a bacterial component that enhances the progression of this disease. And things like diabetes and obesity, we also think that bacteria can be involved in the development of these diseases. Now, it's not all a single one-road direction, but there's also the other way around, right? So we also have a massive influence on the bacteria that colonize us and on the general state of the health state of this gut ecosystem. So there's various factors how we can influence now what kind of microbes are living in us. Among these are the diets that we have, our age actually, physiology, our lifestyle, also like what kind of diseases we have, what our hygiene is, immune system, how active our immune system is, in our general health, and also things like drugs, yeah, and not only antibiotics, but all kinds of drugs have an influence on the bacteria within our gut. So it's super complex, these interactions that go in both directions between our microbiome and ourselves. Now today I would like to talk about 3 studies that we have done in my lab in collaboration with others. These are about the impact of diet on the microbiome. So here we specifically wanted to understand this correlation between diet to microbiome to human health. And the first study I want to talk about, this was together with Maria Tracker and Anthony in my group, Anthony Duncan in my group, did a lot of the analysis here. This was about the impact of dietary bioactives on microbiomes. Excuse me. In this study, we had overall 20 participants from 5 time points. And during the study, they now took a diet that was either enriched in bioactives, so high bioactive, or low bioactive diet. In case you're wondering what are bioactives, these are also called phytochemicals, and it's a bioactive compound produced by plants to protect themselves mostly. But we know that these compounds can have antioxidant and sometimes also antibacterial properties, so they're thought to be good in general for our health. What kind of plants have this? I have a picture here on the right of typical food items that are rich in bioactives, and this was also basically the recommendation we gave to participants. Eat a lot of these kind of fruits and vegetables. Now, just to talk a little bit more about the study design. So we, of course, wanted to ensure that we have a that we have a clean scientific approach. Therefore, we were quite keen of understanding if the macronutrient composition, so the fat and proteins, etc., in the food were changed through our diet intervention. You can see that on the right side. Here we compare now macronutrients in high bioactive in green, low bioactive in blue, and baseline in gray. And you can see that they're pretty much overlapping. So our intervention did not change macronutrient composition, which is great news, right? Because we definitely wanted to avoid this. However, the composition of bioactives massively changes with our two diet interventions. And this again is good. This is exactly what we wanted to achieve. On average, the high bioactive group was probably taking 5 times more bioactive compounds in their diet than they did at baseline. So massive increase in bioactives. Now, what do we see? So do we see any changes in the participants? Well, first we looked at metabolites or metabolomics from the urine and fecal metabolomics. This is both quite useful measures that tell different stories. Fecal metabolomics pretty much reflects what the gut bacteria are doing, how metabolically active they are, so to say. While urine metabolomics reflects quite nicely what the human body is doing, what metabolic processes are happening within our own bodies. The first thing we notice that the high bioactive diet induces much stronger changes than the low bioactive diet. But specifically, urine metabolomics were stronger affected than fecal metabolomics, meaning that probably there's a lot of processes that are happening through this diet intervention in the body, but it seems like much fewer within the bacterial community, within the bacterial ecosystem. Worse yet, if we actually, actually not worse, but better in this case, when we look then at short-chain fatty acids, and short-chain fatty acids are very important end products of bacterial fermentation in the gut, we could actually see that through the bioactive rich diets, certain short-chain fatty acids could be increased. So there were strong correlations between some of the bioactives and short-chain fatty acids, which is great news because in general, if you have higher short-chain fatty acid levels, in general, that means a better gut health. However, when we then try to understand, can we change the gut microbiome community? So can we change which taxa are there and how abundant these different taxa, these different bacterial species are? The news was, the finding was this was actually not so great. So because we could see that most of our bacteria stayed at exactly the same level. There were 2 different species, and I show this here on the right side, where we could see a difference between the high bioactive and low bioactive diet, so that were probably influenced by this diet. But overall, we didn't see many differences. So from the study, the take home a little bit is the evidence points to dietary bioactives changing metabolic and bacterial activity, but the microbiome community overall was not really changed. Now, we were in our next study, we were more interested in understanding what are the short-term changes, right? Previously, in the previous study, this was a 2-week diet intervention. But now in this study, together with Fred Warren and Gary Frost from Imperial and Kasha and Anthony in my group, we were interested in understanding what happens to the small intestinal microbiome when we give different food preparations to participants. The design of the study was that we were giving to all participants the same amount of chickpeas. So we wanted to ensure that we have the same molecular food composition that participants get. But what was changed is that how the food was prepared. So in one case, we had broken chickpea cells. In another case, we had single chickpea cells. And in the last case, we had clustered chickpea cells. So it's just a matter of how much you basically destroy the structure within the food. Participants were getting, we could sample the small intestine of participants through nasopharyngeal intubation that was done with them. And then we took a sample every 60 minutes. And this was a really, really exciting study because the small intestine so far has been very little research due to, problems accessing it, right? So doing a nasopharyngeal intubation is not something very pleasant and easy to do. So these kind of samples are really hard to come by, and correspondingly, we understand very little about the small intestinal microbiome, to be fair. So what do we see in the study? Well, the first thing we see is that the food has a major impact on the small intestinal microbiome, and this changes rapidly, right? It changes within 60, 120 minutes. So on one hand, we see that the alpha diversity, so the overall richness, the number of different species, increases with the arrival of food in the small intestine. On the other hand, we see, and I have this graph on the right side, the very different reactions of the microbiome to these two different diets. I just showed two of the three diets now for simplicity. So you can see here that between diet 1 and diet 2, this is a time on the x-axis, and on the y-axis we have different bacterial species in their relative proportion. And you can see really just from the colors already, it's different species that react to these different food preparations. Interestingly, we see a lot of oral bacteria arriving with the food, and these seem to displace the resident gastrointestinal bacteria. Again, this is something completely new, because so far we were assuming that oral bacteria are not very much passing through the acid barrier in the stomach, so that was quite a surprise for us. Also, we saw that how strong these oral bacteria are replacing the gastrointestinal resident bacteria really depends a bit on the food preparation, and we think this is related to how much the bacteria could actually get into the food and pass, probably protected a little bit in the food matrix, pass the acid barrier in the stomach. Here we used, and to further follow this up, to understand better, or to really be sure that the oral bacteria are replacing the gastrointestinal bacteria, we used strain-resolved metagenomics. So here we go in at a really high genomic resolution, and in this way we can then track oral bacteria from the oral metagenome down until they arrive in the small intestine. And what you see on this graph is the percent of oral strains that are found in the ileum, in the small intestinal samples. And you can see over time, this really increases with the arrival of the food bolus, right? The main food bolus arrives around 120 minutes in each of these cases. And this goes up to more than 50% even for the broken chickpeas case, right? Interestingly, again, depending on the food preparation, more or less oral bacteria were arriving in the small intestine. So to further understand the stability in this ecosystem, we now had a measurement of fecal metabolites and small intestinal metabolites and small intestinal bacteria, the metabolites as boxes, the bacteria as circles in this graph. And we built a co-occurrence network to understand which of these bacteria are often occurring together, which of these bacteria are excluding each other, and how's the relation to metabolites. So here we see a cluster of metabolites, and in the down there you see a cluster of oral bacteria. These are mostly co-occurring, so they kind of occur at the same time, and this makes all sense, especially for the oral bacteria. Interestingly, the metabolites seem pretty independent from bacteria. There's not strong correlations to any of the bacteria, so there's no scenario of one bacteria, one metabolite. This is a very important message, because this means that probably the whole community of bacteria is somehow contributing to these different metabolites being produced and used up and ultimately being made available to the human host. Now, this is the situation at T0, so at 0 minutes in the experiment. So if we go a bit further to 120 minutes, we see suddenly that the correlations have completely changed between these different bacterial taxa, right? So many of the food arrivals really seems to disrupt the established networks between different bacteria, which could probably mean that these bacteria can rapidly change who they're working together with to conduct complex metabolic reactions within the food, through the food. So what's happening here, I think we actually have to kind of rethink the small intestinal microbiome. Right? So what we see from this experiment is clearly that there's a lot of oral bacteria that also contribute to the functioning of the of the small intestinal microbiome. So with each food, you could probably imagine that the oral bacteria here in blue are increasing in their abundance overall in the small intestine, and that with time afterwards, gut bacteria replace again the oral bacteria because they're simply better adapted to this kind of environment. However, at the peak, this is also when a lot of the metabolite turnover within the small intestine happens. So it might be quite important that these two, the oral bacteria and the gut bacteria, are working together on processing our food. So in general, I think from the study we can learn that the oral microbiome is crucial in food processing beyond the oral cavity, and that there's a very dynamic composition of the ileal microbiome. And this is dependent on what kind of food we're eating, right? It's a relative fraction of oral to gut bacteria. Now, last, I want to talk about a study that we did with Janis Bedarf and Ulrich Wüllner in Bonn in Germany, and Anthony from my group was again helping a bit with analysis. So Janis, Ulrich, and me, we have been working for decades, well, for a decade, not decades, not that old yet, yeah, but for a decade together already. And specifically, we have been working on Parkinson's disease and the gut microbiome. And what we found in past studies was Parkinson's disease patients have a dysbiotic gut microbiome. So, for example, we find a lot of pro-inflammatory bacteria or an increase in pro-inflammatory bacteria in this patient group. And we also know that, in general, beneficial short-chain fatty acids are reduced within the Parkinson's disease gut microbiome. So we designed a study where we said, well, let's maybe try with a diet intervention to restore the gut microbiome and short-chain fatty acid levels within the Parkinson's cohort. So this was a relatively small study. This was planned as a pilot study. So we had 10 Parkinson's patients, and we had also 10 controls. These were actually household controls. So these were healthy spouses of the Parkinson's disease patients. And we, for each of these, we asked them to take a 4-week prebiotic diet that would increase short-chain fatty acids levels, at least in theory, within the gut microbiome. And after that, we analyzed our data. So what do we find? Well, there's actually some good news here. So the prebiotic intervention can restore in general metabolic profiles. So can restore the metabolic differences that we see between Parkinson's disease and control patients. Specifically, short-chain fatty acid levels are closer to control after this prebiotic intervention. You can see that on the right side in the graph. These are now 6 typical short-chain fatty acids, different classes of them. And in green, you see the control patients. In blue, the Parkinson's disease patients. And this is before, after. So comparing the levels of these short-chain fatty acids before and after the prebiotic intervention. And you see, especially for the Parkinson's disease patient in this dark blue color here, their short-chain fatty acid levels are often the same as the controls after our prebiotic intervention. So that's good news. We can probably restore this level back to a more normal level for them. Also, we see that GI symptoms seem to normalize. Specifically, disease severity was reduced on day 28, so after the intervention. So that's good news. However, this was not, the disease severity specifically was not significant. So this was only a trend we saw there. But overall, we thought it was quite encouraging. However, when we looked at the microbial dysbiosis, we found that the prebiotic intervention does not restore a healthy gut microbiome, right? So here we compared now the differences in the gut microbiome between Parkinson's disease patients and controls. And we do that before the diet intervention. You see it on the y-axis, and after, and you see it on the x-axis. The taxa that we find, so the dysbiosis of the microbiome, is actually a typical Parkinson's disease dysbiosis. So this is not different from dozens of other studies that have been published on the subject. However, what is interesting here is that after the prebiotic intervention, we actually do see a stronger shift in these taxa, right? So the dysbiosis seems to increase to our diet intervention rather than decrease. We don't really know why that is. And in general, it's quite difficult actually now, at least on our current level, to predict really what the diet intervention will do to a gut microbiome. But yeah, in this case, we see it does not really restore the dysbiosis. However, what we can also look at is which bacterial species actually benefit from the diet intervention. And here we can do this in the control patients. You see it on the y-axis now, and in the Parkinson's disease patients. So that's on the x-axis. And the good news is it's pretty much the same bacteria that benefit from our prebiotic intervention. We see multiple bifidobacteria species increasing in their abundance after our prebiotic intervention. Now, this is nothing new. We know this from other studies that this kind of specific prebiotics we were giving usually benefit bifidobacterium, but it's good to see that this works in Parkinson's as well as in healthy controls. However, there's actually some better news here. So overall, we also find that the prebiotic intervention seems to beneficially change the gut microbiome of these Parkinson's disease patients. So now we were actually looking next at typical metabolites that are known to either be neuroprotective or neurotoxic. And in most cases, these change for the better. So meaning neuroprotective going up, neurotoxic going down. And we can also link this to bacterial pathways that are involved in the production of these metabolites, right? So quininic acid, for example, degradation goes up after the diet intervention. You see that here in the dark blue. And quininic acid levels in the urine seem to at least go down, specifically in controls, but also a little bit in the PD group. So there seems to be some link between what the gut microbiome is doing and what we are seeing happening within the urine metabolomics. So overall, from the study, we think that through the diet intervention, the PD dysbiosis can't be reverted, but the symptoms can be reduced. Furthermore, we see that neurotoxic bacterial pathways and metabolites are reduced and neuroprotective correspondingly increased. Last, I want to just briefly talk about current gaps in our microbiome, specifically gut microbiome knowledge. So I've been working on this for a long time already, and I think microbiome diversity is actually not very well understood. Right? And this is for several reasons. One of them is that the functional annotations we have of genes are often guesswork. So it's very difficult for us to understand what is a bacterium actually doing within the gut microbiome. Furthermore, underrepresented genomes from rare taxa, such as fungi or lower-banded bacteria, are often very difficult to deal with in the data analysis because they're either missing from reference databases or because pipelines are pretty bad at processing these. Furthermore, pangenomes and adaptation of bacteria is completely underestimated, but we do know that there's a lot of evolutionary processes going on even within single individual human participants. But crucially, and I think this is the biggest problem, we still don't know what a healthy microbiome is. Now, we had a research project recently, so this was with Clémence Prior. She is now in Bordeaux, previously in my group as a postdoc, and Rebecca. And they were developing a machine learning algorithm that tried to classify what is a normal and what is a dysbiotic gut microbiome. And I was quite excited, especially about one result from this machine learning algorithm, because it could automatically identify and pinpoint to us some bacteria that it thought were not good. Now you have to understand, we didn't give a list of pathogens to this algorithm, anything like that. So it could pretty much automatically pick out these taxa that you see in the red group here, in the red box, which are all actually known pathogens to humans, and classifying these as not so great for a healthy, stable environment. It was quite exciting. And I just briefly want to talk to you how this works, and I try to really break it down to very simple pictures, basically, what we wanted to kind of achieve with this model. So if you just look at these pictures here, and you might actually be able to guess that 2 of these could be forest and 2 of these could be classified as grassland. So what happened here is we actually used ecosystem types, right? We just gave ecosystem labels to these pictures. And this is a very powerful concept of classifying things, putting things in boxes, so to say, because this actually gives us a lot of information. Yeah. So in the forest, for example, we do know what kind of animals to expect. We would probably also know what kind of food to find, or what might be good clothes to wear, etc. if we go to such an environment, similar for grassland. So using ecosystem types also as a simple label can really give us a lot of information about a certain environment. And you have to now imagine the gut microbiome is in the end an ecosystem. It's no different from a forest, but of course it's on a microscopic scale. So we try to do something similar now with our gut microbiomes, and this is what we call an endosignature model. Basically, here what you see is a fitness landscape of the typical gut microbiome compositions. And if we can only, with machine learning approaches, detect these peaks, these optimal states within this very high dimensional space, that can tell us a lot about typical bacteria that should be occurring in a healthy microbiome, right? So our endosignature model can capture these signatures now of healthy microbiomes, and it gives us a very simplified view of the gut ecosystem, while the underlying machine learning system and algorithms have all the details that they need, so to say. And it allows us to identify dysbiotic microbiomes. If you're more interested in that, we have a publication on this. This was published in 2023. And with this, I'm already at the end of my talk. I want to thank you all for listening, and specifically thanks Marion for the invitation, and Jeremy, and my various collaborators that were contributing to some of this research, my founders, but specifically my group. You see them here. This is us in the Quantum Institute. And yeah, thank you.
Wow. Thank you very much, Hal. Well, that's absolutely mind-blowing, just mind-blowing. And it just highlights how much we don't know. You've been studying this for years and years and years, and it's very humbling to hear you say that we still don't understand the microbiome. So there you go. Well,
Thanks.
thank you. Thank you. That's clearly years and years of your life that you've poured into that. And it's fascinating about the Parkinson's disease, because as general practitioners, we see people with Parkinson's, and one of the big problems that they have is bowel issues, mainly constipation, this sort of stuff. If there's anything that we can apply to that, some practical learning, then that would be very helpful to us. So thank you. That's been very, very insightful. We're going to move straight on, and we'll take questions at the end. I can see a question on the thing, so thank you, audience, for that, and keep them coming. Delighted to introduce now Kevin Barrett. Kevin's a GP in London, but he's very he's got fingers in lots of pies, shall we say. Kevin often speaks on inflammatory bowel disease, but today he's talking about lifestyle medicine, which is a different tack for Kevin. But we're delighted that you're with us, Kevin, and thank you for speaking. So over to you.
Thank you, Marion. Thank you, Falk. Oh gosh, I'm going to find it hard to follow that talk. My brief for today is to talk about what we can do as GPs to give the advice to our patients when they come to us with a whole range of different issues. Topics: lifestyle medicine. Now, I have been to a couple of courses on lifestyle medicine, which potentially makes me more qualified to advise people than some of the people giving that advice out there at the moment. However, my primary talk really is just to have a generalist GP's view on different things. I've been interested in microbiome for a number of years. I'm a patient with IBD, so I've had bowel symptoms for a long time. I enjoy running, and I've had a vegetarian diet since medical school. So I'm going to bring some of those little nuggets to life today. So I thought, what can I do? I can talk about a few different areas. I can't talk about a lot, but what the sort of things the patients come to us and ask us about, and what sort of things are they interested in, and how can we give them those little nuggets of advice. So I'm hoping you'll get some little takeaway things from today. So I thought I would talk about fiber and coffee and ultra-processed foods, and obviously exercise and parkrun, for those of you that recognise the parkrun symbol down there as well. One thing I'm not going to talk about is fecal transplants. There's a lot of interest in that at the moment, and a lot of sort of dark web type industries going on. People are trading their own fecal samples and experimenting with that as well. Very clear at the moment that should only be kept within the realms of scientific studies. So please don't encourage any of your patients to go off and do anything like that. But there may be some promise. We've heard promising things in the past, and hopefully that will come around again in the future for treating a wide range of diseases. So patients come to us and think, well, the gut is just a tube of muscle and poo, isn't it? That's all it does. It just squeezes things along, and you put food in one end, and out comes poo. And we've heard from folks talk that actually things are far, far, far more complicated than that. This slide is one thing that I looked at a few years ago when I was interested in the looking at the pathophysiology and the development of inflammatory bowel disease. Now, we heard about Parkinson's disease and the pro-inflammatory state that can occur in the gut and actually the rest of the body that you get with Parkinson's disease. We see a very similar picture with inflammatory bowel disease, with psoriasis, with rheumatoid arthritis, even with diabetes, that there's a pro-inflammatory state implicated in that. This slide just shows some of the complicated interactions that go on between the gut microbiome, the wall of the enterocytes, and the immune system. The gut-associated lymph tissue is actually one of the largest organs in the body. It's certainly the largest source of immune cells and immune systems. The interaction between the microbiome, the bacteria themselves, the short-chain fatty acids that they produce, the interleukins that sit on the surface, the immunoglobulins as well, how they all interact with each other is fascinating. We used to think that there were good bacteria and bad bacteria. But we know that's not the case now. So it's generally some bacteria we used to think were good bacteria in certain circumstances can have a negative effect, and vice versa. Those bacteria we thought were always going to be bad and have a negative influence, actually in some situations can regulate others, and have a potentially positive impact on health. So it's vastly complicated, and I think we're only just scraping the surface of understanding it, as Falk says. So the reason I showed this diagram as well is to show what happened. There was a theory called the leaky gut hypothesis, which still has some elements of it that are true, where the immunoglobulins and the immune impact on the surface of the gut, in the lumen of the gut, can pass through the gut wall and trigger off a reaction on the lining of the gut, which can influence and particularly inflammatory bowel disease, and trigger off a range of diseases. So bear that in mind for the next slide, because I want to talk a little bit about ultra-processed foods. Now, there's a huge industry going on at the moment to try and get people to have a plant-based diet. Now, there are plant-based diets and there are plant-based diets. And there are processed foods, and there are highly processed foods, and there are ultra-processed foods. The issue with lots of these ones, so particularly the highly processed and ultra-processed foods, is they have a lot of food additives in there. So hydrocolloids in particular are often added. So these are added because they make the food look nicer, they taste nicer, they change the viscosity, the texture, they stabilize the food, they make it last longer, and they help protect it against some temperature changes. So they have positive benefits. However, they can also affect the lining of the gut wall. They can potentially impact some of the protective membranes and measures on the lining, and potentially influence how much of the gut wall is exposed to some of the negative influences. So that's one of the reasons why the emulsifiers and can impact things. Years ago, it was thought that highly processed foods did not have any role to play in the development of inflammatory bowel disease. This was postulated, but was shot down. However, recent years, actually, there's more and more evidence that a diet high in ultra-processed foods can predispose some people to developing inflammatory bowel disease and some of the other autoimmune inflammatory conditions as well. So that physical effect of breaking down that barrier on the surface of the lining of the gut can actually have a role to play. So bear that in mind when we're looking at ultra-processed foods. That includes plant-based diets. I then thought, well, actually, surely we should be talking about reducing meat because of environmental impact, and the land use and all of those negative consequences of that. And that's absolutely true. However, highly processed foods, not necessarily the most nutritious. Apologies for a very simplistic explanation to any dietitians in the audience today as well. But we know they're high in energy. so which is not necessarily a good thing. High in salt and high in fats, and low in the general overall quality of the protein that's in there as well. So they're absolutely fine to eat if you're doing meat-free Mondays, or having a month of being vegetarian or even vegan, and you go out to a chain restaurant and it's one of the options on there as well. It's absolutely fine, but I wouldn't recommend it as a everyday part of your diet. There are different ways of looking at that. So I then thought, if I go into one of our middle of the road supermarkets, some of you might be able to work out which one that this is, but I just chose one that quite a lot of people shop at. So, and I chose 3 different burgers. So on here we've got a sort of meat substitute burger. We've got a real meat burger, and we've got a vegetable burger. Now, looking at these ones, I don't know if people can guess which one's which from here. I'm not going to tell you at the moment, but this is the nutritional content on here. Some are higher in fat than others. Some have got higher saturated fat content. Some have got more salt than others as well. So a good mixture of things on here. So these are just the general 3. It's quite hard to tell which one's which just looking at this, although you might have some clues. And again, I'm sure the dietitians will have a better answer than that. So this is what they are. The one on the left is your soya meat-free substitute. The one in the middle is more of a vegetable burger, and the one on the right is your straightforward, bog standard, value type burger on there. So nothing too special about any of those, but these are ones that our patients will be eating every day. I then looked at the ingredients of all of these. And one of the things I've learned from all of this actually is that some of these foods do have a number of different foods which can contribute to that potential negative impact on the lining of the gut, and the potential for impacting the immune system, the immune response, and inflammatory response that goes on in the gut. And you can see from here actually that although quite a lot of the foods are actually okay, these are not the worst examples by a long shot. And there are certainly going to be worse ones out there as well. There are quite a lot of I'm not going to say unnatural, but suddenly ingredients that might not have been in these foods 20 or 30 years ago. But actually looking at the beef burger, so even as a vegetarian looking at the beef burger, there's not a lot of additive type things in there. It looks like fairly straightforward ingredients within that actually. So, I know it's higher in fat and higher in salt, but actually there's less processed food within that one. So it may not actually be the worst option. So I'm sorry to shock anyone who's vegetarian or vegan in the audience with that, but that's one thing to think about as well. My general rule of thumb when looking at the ingredients of things is that the more ingredients a product has, the less likely it is to be good for you. So that's my rule of thumb with patients. Look at the list of ingredients. If it's a long list with lots of words that are long and complicated, generally try and avoid that one. Go for one that's just made of straightforward, bog standard vegetable type materials in there with a bit of stuff to hold it together. And that's a good rule of thumb to go by. So the shorter the ingredient list, the better. So that's one of my top tips for patients. Other things patients come to us to say, and we had folks saying this earlier on, is that actually there is no established thing as a good or healthy microbiome. Also, depends where you get your sample from. So there are a number of companies out there, I'm not going to name one of the big ones, but there are lots of companies out there who offer to test your microbiome and tell you whether you've got a good or a healthy or a diverse microbiome or not. However, these 3 graphs in here, the sort of peach colour one on the left is what happens if you get a stool sample. So it's the ones we give to our patients in the surgery, the little blue pots with the spoons in. Sorry for those that are still eating their dinner. The middle one on there is ones that are taken at biopsy. So you have an endoscopy and they take a sample from the gut wall. And the third one is the one that you take at endoscopy, so kind of by washout. And you can see there's a difference in there. And some of that makes sense because the the bacteria that we're shedding in our stool, that has perhaps a different population because it's potentially one that there's no use, not as much use for anymore. The ones that are kind of clinging onto the surface of the villi in our guts, they probably have a different role. And there are other ones that are more deeply buried within the wall and actually interacting more closely with the enterocytes and the wall. So all those three populations, there's an overlap. And again, there's some quite wide confidence intervals on this graph, but there's a difference between those different populations. So is actually having a fecal stool sample microbiome, does it actually reflect truly what's going on close to the walls of your gut? And does it actually reflect what's happening? Jury's out on that, but I'm sure we'll find out more about it in the future. However, it's much harder to collect samples endoscopically in our surgeries. Fibre, that's another big topic as well. So adults recommendation from the NHS website and British Dietitian Association is that adults should get around 30 grams of dietary fibre each day. I don't know about you, I find it really hard to picture what 30 grams is. But then I also looked at this and thought, actually, well, what's the average actual intake for adults? So over 16-year-olds in the UK, what's the average daily intake of fibre? Is it 5 grams, 11 grams, 18 grams, 22 grams? Notice I didn't put 30 grams there because we know it's not going to be anywhere near that. It's actually 18 grams. So we're getting just over half the dietary fibre that we should be getting. A whole host of reasons for that. So when patients come to us and say, what should I be doing? What are the simple swaps that you can do? So if they eat white bread, then we go for a half and half, 50-50 equivalent. That's a good starting point. Just these little simple changes. Put a handful of frozen vegetables in. So keep a bag of frozen veg in the freezer and just stick some frozen veg in a soup or a sauce alongside things. Try and swap your white pasta, white rice for brown rice and brown pasta. Try a different sort of bread. Eat a bit more of the skin of the potato. Simple things like that. Interestingly, so kiwi fruit, another favourite topic of mine. Many people actually eat the skin of the kiwi fruit. You don't have to peel a kiwi fruit. You can eat it quite happily with the skin on, and that's a really good source of fibre. Takes a bit of getting used to, but again, lots of simple, straightforward things that you can do that don't cost a lot of money to our patients on limited incomes. So how can we just generally try and get our patients to increase their fibre intake, because that makes a huge benefit to their health. However, dietitians in the audience again will say, what about FODMAPs? So FODMAPs, we should all know about that. It's foods that are fermentable oligodisaccharides, monosaccharides, and polyols. I had to practice that several times. Now these are foods that ferment inside the gut. Producing gas, they make you retain water. And for patients with irritable bowel syndrome, so the functional type bowel disorders, some patients have a greater sensitivity to increased pressure, and their pain receptors to fire off at lower thresholds than others. So that can give you lots of symptoms of IBS. Now, you can get IBS symptoms along with other diseases, inflammatory bowel disease. Lots of patients, about 25 to 30% of patients with inflammatory bowel disease have functional IBS type symptoms alongside it. And it can be quite difficult to tell the difference sometimes between an inflammatory flare of IBD versus a functional flare of the IBS type symptoms. So to be a little bit careful with these ones. So again, keep a food diary. Take things in moderation. There are lots of sources of lists of foods that are high in FODMAPs. You can do single food exclusion diets. So nice guidance on IBS talks about doing single food exclusion diets and keeping food diaries, and that can absolutely work. And you can divide the FODMAPs into those different groups. But again, if in doubt, some people actually need to be referred to a dietitian. Clearly, I'm not a dietitian. I'm not married to one. I don't have any shares in any companies along those lines. But I think dietitians are fantastic, and we need more of them to give the advice, that sort of individualised advice to patients, to support them along their food-related journeys. Anyway, another favourite topic of mine: parkrun. Yes, I like parkrun and running, etc. as well. And everyone bangs on about parkrun these days. Parkrun. Why is parkrun so amazing? Huge reasons for it. Social benefit, fresh air, being outside in nature, being with other people. But actually, if you look at the impact on the gut microbiome, there's no particular studies are found yet that are particularly related to parkrun and the impact on the gut microbiome. I'm sure that some of the universities, there's a university in Hertfordshire, where one of the researchers does quite a lot of exercise related research. But if you look at the impact on the gut microbiome, I chose this study because it's a fairly recent one in a big well-respected journal that shows that increase in bifidobacterium. So bifidobacterium is one of those so-called good bacteria. The more bifidobacterium we have, then generally the better things are, alongside a diverse microbiome, of course. That actually, if you exercise regularly for between 30 and 60 minutes, so that's sort of the average time it takes people to do a park run. Those of you that whiz around in less than 20 minutes, hats off to you. But if you walk around in 60 minutes, not an issue at all. That's exactly what park run's all about. That 30 to 60 minute window seems to be sort of the optimum. And if you look at this paper in Gut, again, the benefits of exercise seem to peak at around that 30 to 60 minute interval. Again, this is a generalization, you have to individualize it to people, but the benefits on gut health, on cancer risk, they all seem to be around about that 30 to 60 minute interval. People that exercise more than 60 minutes, and I get that when I do longer runs, certainly know that you get the runner's diarrhea, and other symptoms. There's lots of reasons for that. It's the reduced gut perfusion, and the blood flow being diverted away from the gut, as well as increased gut transit time, the slowing of the gut that you get with that too. So it's quite common to see people have to disappear off and relieve themselves in the bushes, sometimes the longer runs. But generally that 30 to 60 minute exercise interval seems to be the optimum. So that's why I think parkrun's amazing. Another favourite of mine, coffee. I drink quite a lot of coffee, probably too much coffee, but whole host of reasons for that. There's lots of research done about coffee. I think if you're a researcher, then actually that'd be my dream job to study coffee all day. Now, there's lots of conflicting studies around this. I chose this particular one because, again, it's related to bifidobacterium, and again, it's to do with coffee increasing. So again, this is sort of the 3 to 4 cups per day of coffee tends to actually increase those generally positive bacteria in the gut. Now, the coffee they used in this particular study was ground coffee, so it's freshly ground coffee. Other studies that have used instant coffee found no impact whatsoever. So, is that to do with something to do with the processing, or is it the fact that there's lots of fibre in the coffee itself? And fibre, we know, is a prebiotic. So, is it the fibre itself, or the coffee, or a bit of both? I'm not entirely sure with that yet. I didn't have a lot of time to kind of do a lot more delving into that, but one day, maybe when I'm retired, I might do more research into that. However, we all know if you drink too much coffee, you have this effect on the guts as well. So it's definitely a stimulant, which, again, can be a positive benefit for lots of people, but not necessarily for everybody. So again, that sort of 3 to 4 cups of coffee per day seems to be the sort of the general optimum for that. Again, bearing in mind the negative impact they can have. So my summary really for patients, what simple advice can we give to them alongside fresh air, exercise, nature, all those amazing things we have to do. But in terms of diet, 30 grams of fibre we should be aiming for. If you can't get to 30 grams, any increase is better than normal. Small, simple changes, the little simple, cheap, effective changes can make a huge difference. Try and aim for 30 different fruits and vegetables each week. So the 5 a day thing is true. It's actually technically should be, or ideally 7 for women and 9 for men per day, and 30 per week. But again, anything is better than nothing. So aim for 30 different ones each week. Keep food simple. Again, look at the list of ingredients. The fewer ingredients that are in a packaged food, generally the better that is. That's again a rule of thumb. Coffee in moderation can be good for you, benefits to the liver as well as the gut. Regular exercise, a little bit of something several days a week is important. And last but not least, don't forget about dietitians. If you've got access to one, please make the most of them. And that's it from me. Thank you very much, everyone, for bearing with me today.
Absolutely fantastic. Thank you, Kevin, very much. Wish we could do you a round of applause. I suppose we can't really. Wow. Well, on one hand, it's very, very simple, but on the other hand, it's clearly very complex, isn't it? We've got quite a number of questions in the chat. We've got plenty of time for questions, so if anybody's got any more questions, we'd welcome them. They're grouped, some for Falk, some for you, Kevin. Wow, what a rich learning, especially the lifestyle one. There is a lot of interest, and lots of people think they know a lot about it, don't they? We do the park run in Sheffield. We were one of the practices that set up the park run in our local park, and it has benefit on so many levels. Must admit, I hadn't realised the effect on the microbiome, so that's new to me. But certainly the social benefits, and just you can't measure them, that come from it. But you can translate that into any sort of exercise, and this 30 minutes, 5 days a week is a very good mantra, and as is the 30 different types of fruit and vegetables. But I take your point that people struggle to do 5 a day, and then when they said 10 a day, I think the general public took fright. So let's have a look at some of these questions. They came in order of the speakers. So the first one, Falk, is for you. And it was talking about the Parkinson's dysbiosis study, asking about the time frame, because you mentioned 2 weeks. That's obviously a brief amount of time. They're asking, could you do it for 6 months? And what about a prebiotic rich diet, or what are your thoughts on that?
Yeah, thanks for the great question. So, actually, we had 4 weeks in this one. I think it was in one of the other studies where we had 2 weeks only.
Right. Yeah.
But yeah, we would have loved to do that for longer. However, the impact on the participants was like, well, so because we were giving this really prebiotic rich diet, and maybe I can also kind of go ahead a little bit. There was another question, I think, here about what this diet actually was, or the intervention. So the intervention was to, first off, lactulose, which is a normal probiotic that's also given to Parkinson's disease patients, but this kind of loosens your stool, and that's already known. Yeah, so it's against constipation. But then we also, the recommendation was to eat lots of apples. So it was at least 2 apples per day. And also lots of lentils or beans, so food with resistant starches, 5 portions per week. And in general, I think the Parkinson's disease patients didn't mind this too much, but especially the healthy controls kind of didn't want to do that too long, this diet, because it can lead to bloating. It can lead to also having a higher stool frequency. So yeah, we would have loved to do that for longer, and I think we need to do a follow-up study where we just do that longer. Completely correct question here. But yeah, due to the study design, this being a pilot study and so on, is that we just want to see, can we actually restore short-chain fatty acid levels, was 4 weeks for now.
Yes. A related question a bit further down the list is, what are your thoughts on Movicol? So Movicol is a medication that we do prescribe for Parkinson's patients. Have you any thoughts on that? It's a macrogol, I think. Is that right?
I'm actually not aware of this movie called, sorry.
It's a prescription medicine.
Yeah.
I suppose it's not very physiological. It draws a lot of water into the bowel and retains it there, and helps to propel the contents along.
Okay. So
I mean, basically, it's a laxative, a stool softener and a laxative. But if it's not your field, then it's an unfair question, really.
Well, yeah, so I'm not familiar with that one, but I can, of course, speculate a little on it.
No worries, no worries. Let's go while we've got your brain with us. Let's let's use it. A question from a lady asking about the prebiotic protocol. Was there a specific prebiotic protocol that you used? This is for the PD study.
Yeah, so this was what I mentioned before. So it was lactulose, lots of apples, and
Apples, yeah,
and
and lentils.
yeah, some sort of lentils or beans or whatever, like, so
Right.
stuff.
Yeah,
So we had a list of foods we were giving to them. Potatoes, I think, was also on there. A
yeah,
list of foods, and then make sure you have 5 portions of these per week.
yep, yep. Okay, thank you for that. And then a little bit further down, a question about what about adding friendly, in inverted commas, bacteria like lactobacillus and bifidus with the prebiotics? Would that make things better or worse or different, or what effect does that have on the bowel?
Yeah, so I mean, this is again really good question. But here we're moving from prebiotic into the probiotic
Yeah,
intervention, right? So it's a different kind of intervention you're doing. The problem a bit with probiotics is that we don't really know, like, okay, we can kind of speculate how many of them actually do survive, but often enough, these probiotics might not have really a chance of growing within the gut ecosystem. And if you think about it, like depending on the dosage you take, right? But in the end, if you want to take a probiotic, so a live bacteria, you probably would want that it does something, so that it has some sort of metabolic activity, which means you need to have bacteria that can grow well within the ecosystem. Now, as I explained to you in the talks, is an extremely complex ecosystem with lots of interactions between different partners, and these are like interactions that probably have over years developed to that level of this metabolite exchanges, etc., that's going on between these different bacteria. So my guess would be even if you dump a huge amount of lactobacillus or bifidobacteria into yourself, chances are that a lot of these species don't function that well within the established ecosystem. So
that's fascinating.
my take would be rather take prebiotics, try to increase the abundance of already existing lactobacillus, existing
Yes,
bifidobacteria to higher levels by just giving them the food that is specific to these bacteria.
brilliant, brilliant. That's an interesting answer. Good, lovely. Moving down, thank you. Now then, this is moving into vegan, vegetarian food. And a lady's observing that sometimes it makes patients ill. I suppose this is one for you, Kevin. It says about overall health deteriorates, including mental health.
Or yes, I mean, there are. You can take any diet and have a good or a bad version of it. I certainly agree. If you have a vegan or vegetarian diet that's high in ultra processed foods and low in naturally occurring foods and fruits and vegetables, that's certainly not so good for you. You can get nutrient, vitamin deficiencies, a whole range of different things with that too. So again, the general, my rule of thumb is the simpler the diet, the better. In terms of the number of ingredients that are there, the less processed food, the better. Again, we're all allowed treats, and we're allowed to go out and have nice things from time to time, and I wouldn't stop people doing that at all. But if it becomes, if you take anything to an extreme, it's not good for you. So yeah, just we'll be careful about that. So the fact that somebody says they're vegetarian or vegan doesn't necessarily mean they're healthy. And likewise, somebody who eats nothing but meat isn't necessarily unhealthy either.
Yeah, just going down. Thank you, Kevin. These questions they're fascinating. There's a good one here. Does fasting impact the gut microbiome? I mean, that's relevant at the moment, isn't it? Prolonged fasting.
It is. This is something I've not. So I think my understanding is I think it does, but you have to fast for at least 16 hours, I think, in order to see the impact on that. There's something that's released by the stomach after that prolonged fasting that has an impact on the microbiome. But that's about the extent of my knowledge of it. But it has to be. I think what I remember reading is I think it has to be that's that longer fast to have the impact on it. I still don't entirely know whether it has a longer term beneficial impact. We know that the fasting, the intermittent fastings can have positive impact on weight loss but again, it has to be very individualized and taken in context with the rest of the stuff that's going on in someone's life as well.
Yeah, yeah,
Yeah.
yeah. I think that's right, and everybody's so very different. And I think that Falk, you made that clear at the very beginning that everybody's microbiome is different and individual to them. So that's as far as we can take that one. A point here about fructose corn syrup being pretty bad. I mean, I think we take that as read. Now then, let's go further down. Somebody's obviously attending from the States. I mean, are we good or are we good? That's lovely to see. Research professional athletes get inflammation in the gut, and that the microbiome does change and therefore more gut problems. I mean, Kevin, you talked a little bit about blood circulation to the bowel during exercise, prolonged exercise.
Absolutely. I think the issue is that we know that exercise in the short term creates a pro-inflammatory state in the body, including in the gut. The general thinking is that if you expose your body to a little bit of inflammation on a regular basis, it improves its ability to respond to that and then deals with pathological inflammation in a much better way. Again, professional athletes, they do train, they do tend to have potentially want to lose weight and become as light as they can do to form at their best. So they often have run into problems with nutrition as well. So I wouldn't necessarily take professional athletes as a group to aspire to be. Their careers are often very short, unless you're Paula Radcliffe, who can come back at the age of 51 and run a sub-3 marathon. She's an exception. But yeah, so I think again, everything in moderation is important. But again, keep doing stuff, and that's why that 30 to 60-minute window seems to be the optimum for the majority of people. Yeah,
there is a sweet spot, isn't there, with all of these things for the average punter. We're not elite athletes, are we? Interesting question here about coffee. Coffee with milk, without milk. Does it make any difference?
I think most studies I read were with coffee without milk, because again, just have the dairy again, or a dairy substitute again, just has another complicated factor to it as well.
Yes.
I think so again, lots of people have issues with lactose intolerance to a varying degree. So personally, I have milk in my coffee because I prefer the taste that way. But it's probably not potentially as good as coffee without milk. But that's just my preference.
Yeah. This is a slightly different one. Thank you for that, Kevin. Any research on SIBO? Now, this seems to be the buzz disease of the moment. So, a small intestinal bowel overgrowth. Maybe ask Falk that one first.
Yeah, sure. I can quickly comment. So currently we don't have any SIBO research going on, but again, I think it's one of these overlooked diseases, and it is probably also a bit overlooked for technical reasons. And the technical reason is it's very, very difficult to sample small intestinal microbiome.
Yes.
So if you want to do a study on that, it would be very expensive, let's say, and also requires some really technical knowledge. So yeah. Kevin, anything to add on SIBO?
Having seen a few patients who have either definitely had it or felt that they had it, some have improved with the course of antibiotics. However, it's very difficult to know actually whether it was truly the antibiotics that made the difference for them, and then you get stuck in a cycle where they request more repeated antibiotics, and you run into problems with that. So I try and steer away from that where I can.
Yeah. We're getting down this list now. I've got a good one here. What happens to the microbiome when we have diarrhea? Does it get flushed away? And when will it replenish? Kevin, you go for that one.
Gosh, okay.
Does it get washed out?
You certainly lose more of the luminal bacteria that get flushed away, and probably some of the ones that are on the surface of the villi. With severe diarrhea, and we know people that get the sort of more tropical type diarrhea, that actually you can lose the surface layer of the villi and get temporary lactose intolerance afterwards.
Yes.
So yeah, I'm sure you can do, but lots of them will be clinging on and hiding away, and they're very good at replenishing themselves.
Yeah. Paul, anything to add there? Diarrhea, the effect of diarrhea on the microbiome?
I mean, I guess it's in a way you can compare it to a bowel prep, right, before colonoscopy. And we do know that a bowel prep has actually very little influence long term on the gut microbiome.
Oh, that's interesting. I've often wondered that.
Yeah, so within a month or so, you have the normal,
Yes.
Bowel prep state restored.
Yeah.
Yeah, I would agree with Kevin. So I think the microbiome probably gets replenished from the mucosa.
Yes.
So,
yes.
Bacteria
Just
clinging on there.
developing that argument a little bit. People worry about antibiotic diarrhea and the effect of an antibiotic course on their microbiome. Does it damage it forever, or is it just restored very quickly, or does anybody know that?
Actually, I can comment on that. So I had a study like in 2019 where we specifically looked at this. And in general, if you depends on the antibiotic you're taking, of course. So especially if you take like really broad range antibiotics in high dosages intravenously to top it up, so like really heavy stuff, then you can see an extreme effect on the gut microbiome. So you can really reduce the diversity in the microbiome. And this can last. In our studies, we had a quite extreme case, strong antibiotics intravenously, that lasted for 2 weeks roughly. And then you kind of get back to the more normal level, let's see, but you could still see it over several months that there was a reduction. Interestingly, though, we could see that a lot of the species were restored. So after 2 years, we saw that 3/4 of the original species and specifically also strains were still there. So they were kind of clinging on despite this very, very heavy antibiotic, which was quite amazing to us to see. So yeah, short-term effect for sure. But it probably most of the microbiome is able to survive this.
Thank you. There's a related question right down at the bottom. What about taking probiotics post-antibiotic treatment? Can you just slot that into your answer?
Yeah. So, I mean, probiotic after antibiotic treatment. I'm not sure what I should think of this. To be honest, I'm a bit critical of this. And that is because each time we're taking probiotics, we're potentially replacing bacterial strains within our gut microbiome. Now, the gut microbiome, and this is actually one of the core pillars of my research, is how long does the gut microbiome persist? It persists very, very long, decades. So we have actually quite a few bacteria that we probably inherited from our mothers in our gut microbiome. If you take antibiotics, for example, this is actually these mother-inherited bacteria. That are the ones that you have the highest chance of losing due to the antibiotic course. If you then on top bring in a competitor, so a new bacteria through the probiotic, which is definitely not the strain that you get from your mother, right? It's some industrially, whatever, sourced strain there. There's a very high chance that in the weakened state, your original strain can be replaced. So it's already kind of damaged and, kind of brought down in its abundance due to the antibiotic. So it's kind of receptive for competition. And you might actually replace your naturally, developed symbiotic relationships by taking probiotics. So that's why I personally would be a bit critical, but I also have to say I don't think there's any formal research that is trying to figure out whether this is damaging or not long term.
Yeah, thank you. Yeah, good answers. Very complex, isn't it? You upset the balance of lots of things. Now, there's an interesting little cluster of questions further down. Talking about probiotics from fermented foods or synthetic products. Now, maybe, Kevin, this is one for you. We talk about kimchi and kombucha and fermented foods. and that's one group, or something in a capsule that you swallow in the morning and feel virtuous all day because you've had your dose. Is one more beneficial or effective than another?
So again, I mean, I'm not a researcher into this area at all, but again, the reading I've done around this is that there's more evidence for fermented foods than there is for prebiotics, and there's more evidence for prebiotics than there is for probiotics. So that's my general sort of hierarchy with this as well. The fermented foods, again, there's a difference between the live foods that have lots of fibres within them and lots of bacterial colonies, and the fermented foods that have been pasteurised. So you can buy fresh kombucha, you can buy bottled kombucha, and one's been pasteurised and one hasn't been. So again, you'll get different benefits from those. Again, it varies from which to which. If you look at the populations that drink a lot of naturally fermented foods, sauerkraut and kefir, it may be contributing to general population health within that. Yeah. In terms of the capsules of the probiotics, it's interesting. Again, I read somewhere that actually, as Falk was saying, in order to if you consume however many million bacteria through a drink or a capsule, only a very small percentage of those are going to make it all the way through the acidic gut environment into the gut, and then so you've got to consume huge numbers to have an influence on that. There's also some thinking that is actually, even if the bacteria are denatured or killed as they pass through the stomach, is it the immunoglobulins that are on the surface of the bacteria that have the effect on the immune system and the interaction with the rest of the bacteria? Is that how they work? So again, lots more questions to be answered about that. So yeah.
Oh, the whole thing's fascinating. Yeah, it is. Going back now, thank you, to Falk, there's a question about the study with chickpeas. What's the take-home message in terms of beneficial effect of whole chickpeas versus crushed chickpeas?
I mean, just based on the study, so our study question was not really to find out whether there's any benefits to either of these. We were kind of trying to take a more neutral viewpoint and saying, is there a difference at all, right? Because we knew it was exactly the same macronutrient composition. We just broke open some cells or didn't, so to say. So I wouldn't say that based on what we saw in the study, I would recommend one over the other.
Yeah. Okay, so it ended up the same place in the end. Lovely, thank you for that. Just changing tack a little tiny bit now. There's a couple of questions about ileostomy. How does only having a small bowel affect the microbiome? You haven't got a colon, so you've only got the small bowel because you've got an ileostomy. Is there any implications for long-term health because of that? I mean, maybe nobody knows. And they wouldn't persuade many people to have an ileostomy if it was thought to be detrimental, would they? But so that was a good question.
Yeah, very good question. I mean, in terms of I'm sure there must be some studies around, but I didn't read up on this. But just to speculate a little bit, so if you're missing your colon, you're basically missing a lot of steps in the uptake of nutrients and water resorption, etc., from your diet, right? So there must be definitely a lot of obvious disadvantages from that. But we're just considering the gut microbiome, you're basically missing the final step of the fermentation of food. So everything that would produce short-chain fatty acids is probably reduced. Now we know that short-chain fatty acids are actually quite important for gut health. So they, for example, can calm down the immune system a little bit to avoid inflammation. But they're also important for well-being. So they're also related. Short-chain fatty acids have been linked to mental health. So I think there could be several routes where this definitely disadvantages people not having a colon.
Yeah. Sticking with the bowel, the question about COVID and the effect of COVID on the bowel, and is there any thoughts about manipulating the microbiome to be beneficial to the recovery from long COVID?
Shall I have?
Thank you, Kevin.
Sorry.
Thank you, Kevin.
It's a lot of unexpected questions, which is it's just fascinating. I think again, I'm listening to a set of podcasts that were launched at the beginning of COVID, and just kind of reliving the whole COVID journey again. With hindsight, it's been fascinating, and listen to the research that's coming out, and that we know that COVID is a multi-system inflammatory disease process, particularly affecting the vasculature. So the impact on the gut absolutely is true. It was postulated we would see a rise in autoimmune conditions as a consequence of the COVID pandemic, but I've not seen that translate into reality yet. I'm not entirely sure why, because of the potential triggering of patients at risk of autoimmune conditions. Again, we know that patients with long COVID certainly have GI effects as part of a group of syndromes that they can sadly suffer from that. In terms of what to do about it, I don't know of any research that anyone's done for it. I mean, again, the same general advice about having a high fiber, varied diets is probably going to be absolutely as valid and as important for that group of patients as it is for everyone else, really. That's probably the best I can come up with. Sorry.
Yes, no, I mean, this is where we're at, which is why it's such a fascinating area to talk through, especially when we've got a very rich resource in you and Falk here this evening. Falk, you mentioned the oral microbiome going down. There's a question about using Oral-D or like a mouse dental freshener thing. Does that have any detrimental effect on the microbiome?
So I mean, I think with these these mouthwashes in general.
Mouthwashes in general, yeah.
I think in my field, we're dealing a lot with the hygiene hypothesis. I don't know how familiar you are with this, but basically there's this hypothesis that if you do too much washing of your hands and too much, cleaning of babies and so on, this is detrimental for the immune system, because the immune system is basically getting bored and starting attacking the own body. To just very briefly, probably imprecisely summarize this hypothesis. And I can imagine we have similar cross effects happening with mouthwashes, right? So if you just take too much of that, you probably destroy the microbiome that is in your mouth. In fact, as far as I remember, there's also there's even warnings on the bottles, right? If you take too much mouthwash, there's fungi that can suddenly grow in your mouth and stuff like that. So because you destroy the natural bacterial microbiome, maybe a bit too much, and there's certainly some pathogenic effects that can occur, right? So I think I would view this like the hygiene hypothesis. Maybe don't wash your hands every 10 minutes, maybe in more regular intervals, and probably the same with mouthwashes. So
Yes, it's that balance, isn't it? It's upsetting the balance. Kevin, there's one for you here. Rifamycin, given for SIBO or gut dysbiosis. Does it kill all the good bacteria too?
The short answer is no, but it's not the worst one for affecting the gut microbiome. The trouble is what it's. It certainly will have an impact. But the gut will recover when it's stopped. Again, whether it takes 2 weeks or 10 weeks or 6 months to recover, I'm not entirely sure how long that will do. But but it will have an impact while you're taking it. Absolutely.
Yeah. Thank you for that. Probably one last question, and then it's nearly time to wrap up. Thank you for some very good questions. What an amazing evening we've had. So the last question is, and we'll take Falk first, microbiome and aging. It's like talk for 1 minute on microbiome and aging.
Right, microbiome and aging. So, I mean, there's been quite a few interesting research going on there. And for example, my colleague Amy Parker and Simon Carding at the Quantum Institute, they're quite active in this. What do we know about microbiome and aging? Well, we do know that the older you get, the less control the immune system probably has over the microbiome, over the gut microbiome specifically. And this is also related to processes like inflammaging, yeah? So like an aging-associated inflammation. The microbiome gets less well defined in the composition, the bacteria that are there. You can imagine it a little bit like a forest that is very standardized, and you have exactly the animals and the trees and the plants that you expect, compared to a forest where you suddenly have some really strange invading plants cropping up here and there, and you kind of have the feeling this doesn't look like a proper forest any longer, like a healthy forest like I know from the movies, kind of. Right? And I think it's a similar situation in a way you could imagine with the aging microbiome. So it is kind of diverging into states that are not so normal. We know that there's often also pro-inflammatory bacteria, and in some cases, specifically with people that are like in residential homes, so that are not so mobile anymore, we see a clear deterioration of the microbiome that then correlates to inflammation often enough.
Yeah, thank you. Right, Kevin, no challenge here. You've got 2 minutes. Microbiome and aging.
Well, we absolutely know the microbiome changes throughout life, and some of as Fox said, some of the bacteria that you're born with and you get from your mother probably stay with you throughout your life, and it does change. There are various phases that one goes through, and certainly the sort of midlife hormonal changes, menopause, etc., that can have an impact on that as well. Again, how much of that is caused by aging, and how much of the aging is caused by microbiome, nobody entirely knows. Again, it's that correlation does not imply causation, golden rule that we always go by. I think we know that there are people who might be chronologically old, but actually have a very healthy body, and other people who are actually older than perhaps their chronology might be. And how much of that is to do with their gut microbiome influencing their behaviour? How much of that is to do with their diet and exercise? I wouldn't like to answer that. I think genetics also play a part with that too. So, we should all try and stay as young as we can for as long as we can.
Fabulous. Thank you. Really good way to finish up the evening. Well, what an evening we've had. Thank you very much to our speakers. Thank you, Falk. Thank you, Kevin. We've had some amazing ideas kicking about, and clearly the audience has been very animated and interested and keeps throwing in the odd curveball question. So, it's kept us all on our toes. Thank you to Quadrum Institute. Thank you to Precision Antibiotics. Thank you to the PCSG for hosting it and making it happen. And we're very grateful, and we're bang on 8 p.m. Thank you for your attendance, and we hope to see you next time when we talk some more about the microbiome. Good night.